Our cells are bustling factories, constantly producing the molecules necessary for life. This intricate production process, where genetic information encoded within our DNA is used to create functional products like proteins, is known as gene expression. Think of DNA as a vast library filled with blueprints. Each blueprint holds the instructions for creating a specific protein. Gene expression, however, is a highly regulated process. Cells don't need to make every protein all the time. Just like a library needs a librarian to find the right book, our cells need a system to locate and activate the specific genes required at any given moment. This is where promoters and consensus sequences come into play. These crucial DNA elements act as the librarians of our genome, guiding the cellular machinery to the correct genes and initiating the protein production process. Understanding how promoters and consensus sequences work together is key to unraveling the complexities of gene regulation. Imagine a promoter sequence as a start button located on a piece of DNA. This button, however, isn't a physical button, but rather a specific sequence of DNA nucleotides. Promoter sequences are located upstream, meaning before the coding region of a gene, which contains the instructions for building a protein. When a cell needs to make a specific protein, the molecular machinery responsible for reading DNA and producing proteins, including an enzyme called RNA polymerase, binds to the promoter region of that gene. This binding event signals the start of gene expression. Promoter sequences are essential because they determine where transcription begins. Transcription is the first step in gene expression, where the DNA sequence of a gene is copied into a messenger RNA, mRNA molecule. This mRNA molecule then carries the genetic information from the DNA in the nucleus to the ribosomes in the cytoplasm, where proteins are synthesized. Within promoter regions, there are often short recurring patterns of nucleotides called consensus sequences. These sequences are like common binding motifs for proteins involved in transcription. One well-known consensus sequence is the TATA box, named for its sequence of TATA-EA. Consensus sequences are important because they provide a recognizable binding site for proteins involved in initiating transcription. For instance, the TATA box is recognized by a protein called the TATA binding protein, TBP, which is part of a larger complex that helps position RNA polymerase correctly at the transcription start site. Variations in consensus sequences can affect the binding affinity of transcription factors and, consequently, the rate of transcription. A strong consensus sequence leads to more efficient binding of transcription factors and higher levels of gene expression, while a weak consensus sequence results in lower levels of gene expression. The interplay between promoters and consensus sequences is crucial for precise control of gene expression. The promoter acts as the initial binding site for the transcriptional machinery, while consensus sequences within the promoter provide specific binding sites for transcription factors that fine-tune the process. This intricate interplay ensures that genes are expressed at the right time, in the right cells, and at the right levels. Dysregulation of this process can lead to a myriad of diseases, highlighting the importance of these DNA elements in maintaining cellular function. For instance, mutations in promoter or consensus sequences can lead to altered gene expression and contribute to the development of cancer. Understanding the intricacies of promoter and consensus sequence interactions is crucial for developing targeted therapies for such diseases.